Hello friends, good afternoon and welcome to EduSat live lectures. Dear friends, today in the series of Immunology, we will be discussing about antibody structure and functions. To discuss this topic, we have with us our subject expert, Dr. Hardeep Kaur. Dr. Kaur is Assistant Professor in Department of Zoology in Ramjas College, TU. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
Thank you, Amrit. So dear viewers, we have today the antibody structure and function. This is the topic of our today's discussion. As you all are aware, and I am sure by this time you are aware that there are lots of pathogens around us. Any individual is surrounded by so many pathogens, by the air we breathe in, the food we eat, anything, whatever the drinks we are taking, all these things are having pathogens. Now how to combat those pathogens? Antibody is one protein which is present inside our body which is responsible for taking care of these pathogens. As in our earlier lectures we discussed that the immunity which is produced in our body is of two types, the adaptive immunity is of two types, the cell mediated immunity and the humoral immunity. Now antibody is a very important component of the humoral immunity. Humoral, if you remember, humor meant the body fluid, and so these proteins are actually present in the body fluid. And whenever there is a pathogen, they give a response, an immune response which is very specific. We also discussed in our previous lecture that any antigen will have numerous epitopes or antigenic determining regions. These antigenic determinants, each antigenic determinant will have a specific antibody produced against it. And this antibody will have a specific antigen binding site which will bind to a particular epitope on the antigen. So thereby we can say that an antigen having numerous epitopes can get bound to numerous antibodies. So what are antibodies? Let us understand this. So let us define antibody first of all. An antibody, also known as an immunoglobulin, is a large Y-shaped protein produced by plasma cells. They are recruited by the immune system to identify and neutralize pathogens such as bacteria, viruses, etc. The antibody recognizes a unique molecule of the harmful agent called an antigen, or as we had discussed previously, an epitope or an antigenic determinant region of the antigen. So the antibody comes and binds to this particular epitope, which is, so rather this is a specific antibody binding to a particular epitope. Now where exactly these antibodies are located? As I just told you, these antibodies are produced by the B lymphocytes. Now B lymphocytes can become plasma cells or they can become the memory cells. The plasma cells are antibody producing factories. Now antibody in a B lymphocyte can be present in a membrane bound form or it is present in a secreted form in the blood. It shares structural features and binds to the antigen and participates in number of effector functions. Now as I just told you, any antigen will have numerous epitopes. Numerous epitopes will elicit the production of various different specific antibody to itself. Now are these antibodies different? Yes, they are different vis-a-vis the antigen binding site, but predominantly the rest of the antibody have structural similarities between each one of them. Now their main function is to bind to the antigen, and also once this antigen antibody complex has formed, many effector mechanisms are induced. Main function is the antibody will bind to an antigen. Then it will elicit an immune response wherein many other effector mechanisms, be it opsonization, be it complement fixation or ADCC, that is antibody dependent cell mediated cytotoxicity. What later happens is dependent upon what antibody is binding to a particular epitope on an antigen. It is collectively all antibodies, we call them as immunoglobulins. They do not kill themselves, they just bind to the epitope of an antigen and lead to effector mechanisms which finally leads to killing of an antigen. Now, coming to the history of an antibody, it was A. Tiselius and E. A. Cabot in 1939 who actually found out that immunoglobulins are present in the gamma globulin fraction. Now, what they did was they did a centrifugation, density grade centrifugation, in which the alpha, beta and gamma globulin fractions of the proteins were isolated, and each one of them were tested for immune response, for checking whether they are leading to some immune response, and it was found that the antibodies or the immunoglobulins are actually present in the gamma globulin fraction. Hence, another name for immunoglobulin is also called as gamma globulins. Now, later in 1972, it was Rodney Potter and Gerald Edelman who deciphered the structure of immunoglobulin G, that is IgG, and they got a Nobel Prize in 1972. Now, antibodies belong to immunoglobulin superfamily. Now, what happens is, if once we understand the structure of immunoglobulins, you will see that it has got certain chains which have certain domains. These are because of certain bondings present within the structure. Now, this chain, alpha chain, beta chains and the domain structure, this structure is not only unique to immunoglobulins only. This structure is also present in many surface receptors of T cells and also in certain other molecules like major histocompatibility antigens, etc. Now, wherever this structure is present, we say that because of the similarity in the structure of the protein, all these proteins can be put together in one superfamily. Now, immunoglobulin being the most important component of this superfamily is hence called immunoglobulin superfamily or IGSF. So, in an immunoglobulin superfamily, all molecules have similar structures with disulfide conserved bridges spanning around 70 to 100 residues in length. The major important examples in this immunoglobulin superfamily involves, of course, the antibodies, the T cell receptors, the class 1 and 2 MHC molecules, and also the part of B cell receptor molecules. Now, most members of immunoglobulin superfamily, they cannot bind antigen except for the antibodies or immunoglobulins. Rest of them are not actually may be binding, may not be binding the antigens directly. So, for example, when I am talking about the T cell receptor, they are not wholly binding to an antigen as such. They are just binding to a processed part of an antigen. So, except for immunoglobulins, which whole soul is binding to an antigen, rest of the molecules of a superfamily, immunoglobulin superfamily, may or may not be binding to an antigen. Now, coming again to this immunoglobulin superfamily, as the figure shows that these are the various examples of immunoglobulin superfamily. You can see IgM is given as an example here, immunoglobulin M, T cell receptor, Ig alpha or Ig beta heterodimers, T cell receptors which are also called TCRs, the MHC molecules, that is major histocompatibility molecules, the class 1 as well as the class 2. As you can see, the structure on all these examples is almost similar. It's got linkages, it's got domains, it's got loops formation. So, this similarity in the structure has led to put all these molecules in one superfamily, which we have defined as immunoglobulin superfamily. Now, coming to the antibody structure, which is the main part of discussion of our today's lecture. If we see the antibody structure, we will find that antibodies or immunoglobulins are heterodimers. That is, they have two light chains which here are shown in the green color and two heavy chains. The light chains are almost always identical. It is imperative that they are always identical. They are about 25 kilodaltons in molecular weight, while the two heavy chains, which are again identical in a particular antibody molecule, are about 50 kilodaltons. Of course, this molecular weight is not always final, it depends from which immunoglobulin we are talking about, but when we are defining the generalized structure of an antibody, we will go by this. So, the light chains have a molecular weight approximately 25 kilodaltons, while the heavy chain is approximately of 50 kilodaltons. Now, the heavy chain has been shown as the red chain. Each light chain is bound to the heavy chain by disulfide linkages. As you can see, the yellow interchain disulfide bond, it is actually SS bond. Now, is it the only bond which is present or which is responsible for binding the heavy chain to the light chain? No. There are many other non-covalent interactions like ionic bonds, hydrogen bonds, etc., which are responsible for binding the light chain and the heavy chain. So, technically the covalent interaction due to SS bond, that is disulfide bond, as well as the non-covalent interaction due to ionic bonds, hydrogen bonds, all are responsible for binding of a light chain to a heavy chain. Now, besides that, you will see in the diagram, there are intra-chain disulfide bridges also, like for example, if you see within the heavy chain, if you see there are yellow structures which is the intra-chain disulfide bridges, which is actually giving a loop formation. I mean, if these SS bridges are present within the chain, the rest of the molecule, the rest of the amino acid fragment will get looped, because the SS which is far off, let us say 80 amino acid far off, and then they are forming SS bond, the rest of the molecule will loop out. This looped structure is very similar in most of the molecules of the superfamily. So, intra-chain disulfide bridges gives the loop structure which is called the VL, VH, CH, CH1, CH2, CH3 or CL loops with structure of the antibody molecule. Now, if you see carefully, the first 110 amino acids of the amino terminal of both heavy chain and the light chain forms the variable region. In the heavy chain, this 110 amino acid loop or domain is actually the variable part, which is defined as VH, while in the light chain this 110 amino acid looped or domain is called the VL. Following that, you will see in a heavy chain the rest of the molecule, let us say 330 amino acid long molecule in the heavy chain will form loops of CH1, CH2 and CH3, while in the light chain the 110 amino acids will form the CL region. So, C stands for the constant region while V stands for the variable region. Now, if you see again clearly in this diagram, the half of the heavy chain and the complete light chain is actually forming the FAB fragment, which stands for the fragment where the antigen binding takes place or the antigen binding fragment. So, actually antigen binding is only because of the VH and the VL domain. It is only the 1/4 of the heavy chain and half of the light chain which is responsible for binding to the antigen. But if you go by the nomenclature, the FAB fragment is half of the heavy chain and the complete light chain which is called the FAB fragment or the antigen binding fragment. The rest of the heavy chain, if you see CH2 and the CH3 domain, that is the C terminal half of the heavy chain, is actually called the FC fragment, the fragment that crystallizes. Now, what is the function of this FC fragment? FC fragment has actually responsible for other functions or the effector mechanism. It is the place, as you can see in the CH2 domain, the carbohydrate molecule is actually coming and binding. We all know that antibodies are glycoproteins. So, where is the carbohydrate binding? It is in this CH2 domain that the carbohydrate is binding. Now, this FC fragment, as I told you, has other functions. What are the functions? It is the place where the complement fixation is taking place. We know that complement are certain proteins which have certain effector mechanism, protective mechanism, immune response. It is very important proteins of the immune response. This is the place in the FC fragment where actually the complement fixation is taking place. It is again the region which is responsible for ADCC, that is antibody dependent cell mediated cytotoxicity. So, technically, once an antigen binds to the antigen binding site of an immunoglobulin, the effector mechanism, it depends on the what is binding on the Fc fragment of the antibody, which leads to final killing of the antigen or whatever happens to the antigen. So, the effector mechanism is dependent on the Fc fragment of the antibody. Now, again the same structure of the antibody again shown. Here again there are intra-chain disulfide bonds within each domain also. Now, there are heavy chains, as I just told you, 1 VH and either 3 or 4 CH1, CH2, CH3, CH4 in the heavy chain, and in the light chain we have 1 VL region and 1 CL region, that is 1 variable region and 1 constant region. Now, as you can see in the diagram, there is a hinge region. It is like a hinge of a door. It is rich in proline residue. This because of the presence of the proline residue, this region is highly flexible, and it is also the site of enzyme digestion, as you will see later. Hinge is found in IgG, IgA and IgD, but it is absent in IgM and IgE. It is rich in cysteine residues which are responsible for the disulfide bonds. Now, there is a CDR or the complementarity determining regions present in an antibody, specifically in the variable region where the binding with the antigen takes place. The remaining regions are almost similar within a same class. For example, if I am talking about IgG-A1, let us say, we have variation in the variable region, but IgG-A1 of one type, I mean which is binding to one particular epitope, and the other IgG-A1 which is binding to another epitope, otherwise it will be similar on the constant region. It will only be differing in the variable region which is responsible for its binding to epitope 1 or to epitope B. The rest of the region within a class will be exactly similar. Now, initial experiments, few experiments were done to decipher the structure of antibody. One of the experiment was digestion of an immunoglobulin with papain. This was done by Porter in 1950s. In this case, what happened? By digesting this protein, see papain is a proteolytic enzyme. By digesting this immunoglobulin by papain, we obtained 3 fragments. There were 2 identical Fab fragments and 1 Fc fragment. Now, as I just told you that Fab fragment is the one where antigen binding takes place. So, by having 2 Fab fragments, these are monomeric fragments, the binding would be monomeric, they will bind to each single epitope. The Fc fragment is a distinct fragment, which is the fragment that crystallizes by papain digestion. In following that, another digestion was done by Nizanov in 1950s by pepsin. Pepsin is another proteolytic enzyme, but it has a different site for enzymatic digestion on an immunoglobulin molecule. As you can see, by pepsin digestion, we got Fab2 fragment, which is still wherein the Fab fragments, the half of the Fab fragments, the 2 Fab fragments are joined together by the disulfide linkages. So, we have Fab2. The earlier with the papain digestion, it did not lead to a formation of a lattice with the antigen. It could bind antigen monomerically, but the fragment which we got after pepsin digestion was even able to form a precipitate, because the Fab2 fragment has a complete antigen binding site. Now, in this case, however, the Fc fragment was not recovered, it underwent digestion. There was another experiment done wherein the entire immunoglobulin was reduced by mercaptoethanol treatment, which eliminates disulfide bonds, and finally we will get the H chains and L chains separately. Now, by all these experiments, scientists were able to find out the exact structure of immunoglobulins. Now, there were certain facts about immunoglobulins, that is, immunoglobulins can be secreted or they can be membrane bound. Membrane bound differ in their carboxyl terminal end. There are extracellular spacer of 26 amino acid in the membrane bound, while this spacer is absent in case of the secreted ones. The hydrophobic transmembrane sequence is present, as well as the cytoplasmic tail is present. In addition to binding to the antigens, the antibodies can also promote phagocytosis, as I just told you. they can actually go and opsonize. Opsonization is like a coating, you know. So, if you have a bitter medicine, you do a sugar coating of the medicine. So, similarly, if an antigen comes in our body, the antibody forms a coating of an antigen. This process is called opsonization. By forming an opsonization complex with the antigen antibodies complex can lead, this entire complex can go towards a phagocyte, or a macrophage or a phagocyte can be brought near this complex, and finally it is being killed, the antigen is being killed. Now, this process of coating an antigen is called, maybe to make it more palatable, it is a very colloquial term which is used to make an antigen more palatable, antibody coating is done. So, opsonization is a very important part of antibody molecule. Another thing is, as I told you, the Fc region is also important for other effector mechanisms. For example, complements are a group of proteins which are present which have certain effector mechanisms. Now, once an antigen and an antibody binds together, it leads to activation of certain complement proteins. Once these complement proteins get activated, they lead to further effector mechanism, the entire complement process takes place, and finally the antigen is being killed. There is another method in which is called ADCC, that is antibody dependent cell mediated cytotoxicity, where the antigen antibody is bound. This bound complex goes towards let us say NK cells, which are natural killer cells. So, technically antibody is forming a sandwich between an antigen as well as an NK cell, and finally the NK cells kills this entire complex. Some antibodies can also cross epithelial layer to be excreted through mucus or across placenta. We will come to the later details about antibody in our latest next lecture. Thank you so much. Welcome back, viewers. In our previous lecture we had done the antibody structure. Now in this lecture we will come to the classes of various antibodies and specifically what is monoclonal and polyclonal antibody. As you can see in the figure, friends, the antibody antigen interaction is a very specific interaction. As it is shown, the smaller antigens will fit in the pockets in the variable region of the antibody. In our previous lecture, we had discussed how antigen and antibody combine. If the antigen is very small, it will be fitting in the in pocket of an antibody in the antigen binding site of the antibody. However, if the antigen is big, it will form in an undulating manner where there would be protrusions and depressions, but it would not be like fitting in a pocket. It will be like on a flatter surface or a undulating membrane on the variable region of an antibody. Now, in this particular case, as you can see here, the antigen is actually going inside the pocket formed on the antigen binding site of an antibody. Now, coming to the classes of immunoglobulin, the sequencing of several immunoglobulins had led us to understand the 100 and 110 amino acid terminal is there, which has various hypervariable regions. These hypervariable regions are leading to CDR, that is complementarity determining regions. Now, complementary to epitopes that they will bind. This region is responsible, which gives a complementarity to the antibody. That means the variation present in this variable region would be responsible or for giving specificity to a immunoglobulin molecule to a particular epitope. Now, based on all these sequences, 5 basic sequence patterns have been found in immunoglobulins. The constant region of the heavy chain of immunoglobulin could be of the type alpha, gamma, delta, epsilon or mu. Henceforth, the type of the immunoglobulin would be called IgA, IgG, IgD, IgE and IgM. The above classes, these IgA, G, B, D, E, M, they are all isotypes. That means the difference between all these classes of immunoglobulin is in the heavy chain domain, is in the heavy chain. So, the sequence of the heavy chain, which can be of 5 types, are the 5 isotypes. Now, each class can also have either the kappa or the lambda light chain. That means the heavy chain region of the light chain could be either kappa or could be lambda. Now, any immunoglobulin can have both kappa or both lambda in the light chain. It has been found that in human beings, 60% of the immunoglobulins have kappa light chains and the rest of the 40% immunoglobulins have the lambda chain. While in case of mice, it is 95% of the immunoglobulins have kappa light chains, while the 5% only have the lambda light chains. Now, further we can say, so we said that immunoglobulins are belonging to 5 classes: IgM, IgG, IgA, IgD and IgE. Now, each one of them, for example, IgA and IgG can further be subdivided into subclasses. IgA is divided into IgA1 and IgA2, while IgG is subdivided into IgG1, G2, G3 and G4. You should understand that within a subclass, the IgG1 is definitely different from IgG2, but it is more similar vis-a-vis if I am talking about IgA. So, IgG1 is similar to IgG2 more than it is similar to IgA. So, henceforth IgG1, G2, G3 and G4 belongs to one class IgG and IgA1, A2 is belonging to one class IgA. Now, let us understand, this is the structure of immunoglobulin again. Now, the gamma, delta and epsilon, it is that means giving rise to IgG, IgD and IgA have this structure wherein it has VL region, VH region, CH region, CH2, CH3 and VH regions. So, in the rest of them it is much different. Here we come to the various classes of immunoglobulins. Now, if you see, we have IgG, IgD, IgE, IgM and IgA. As you can see, IgA is a dimer, that is, it is generally present as 2 monomers joined together to form a dimeric IgA. On the other hand, IgM is a pentamer wherein 5 molecules of IgM joined together to form a pentameric molecule, while IgG, IgD and IgE are all monomers in nature. Now, coming individually to each class of the immunoglobulin, let us come to first IgM. IgM antibody is almost, as I just told you, it is pentameric in nature. It is about 5 to 10% of the serum, it is present in 5 to 10% within a serum. It is monomeric. IgM is generally expressed on the B cells, that is B lymphocytes, while if it is secreted, it is in a pentameric manner. That means 5 monomeric IgM will join together and will form a pentameric IgM. It is the first class of antibody produced in the primary response. That means once there is an antigen which comes inside a body, the first antibody which is produced is IgM antibody. It is very good at activating complement and as well as it causes an agglutination reaction. As you can see in this slide, the structure, the C3 molecule and the C4 molecule are involved in forming the pentameric structure. Besides that, there is a presence of a J chain which is leading to the complete pentameric structure. The main function of the J chain is to allow IgM to transport across the epithelium. Now, the molecular weight, because of the presence of 5 molecules or 5 monomeric molecules of IgM, the molecular weight of the pentameric IgM is around 9,70,000. It is the sedimentation coefficient is 19S, and as I just told you, it is the antibody which is produced in the primary response, hence very important. But definitely its concentration is not that much because it is a primary response antibody as compared to IgG, which is a secondary response antibody. Now, coming to IgA. Now, IgA is composed of almost 10 to 15% of total serum immunoglobulins. It is predominantly present as a secretory antibody. As you can see, the 2 monomers are joined together in a dimeric form, and again there is an involvement of the J chain. The J chain and the secretory component helps this dimeric immunoglobulin in transporting across intestinal wall. The J chain makes IgA more resistant to acids and enzymes found in digestive tract. As we all know that immunoglobulin A, that is IgA, is the one which is secreted in the first 3 days after parturition through the breast milk. Now, the breast milk, that is colostrum, which has predominance of IgA, provides the first line of immunity for the neonate. Now, it gives a passive immunity, the naturally occurring passive immunity to the neonate, and hence this IgA is very important. This IgA is present not only in the first 3 days, but also for a long time while the mother is feeding the baby. So, IgA is extremely important. Now, IgA is the main antibody which is going through the digestive tract. Now, digestive tracts have lots of proteolytic enzymes. Now, because of the presence of the J chain, this IgA, the secretory antibody IgA, becomes highly resistant to acids and enzymes which are found in the digestive tract. As already said, since it is a monomer, 2 monomers joined together as a dimer, its molecular weight is like 3,85,000 and the sedimentation coefficient is 11S. Now, coming to the structure of secretory IgA, the secretory IgA, as I just told you, has a J chain and the secretory component which are involved in binding the C3H and the C4H domains of the 2 monomeric IgA molecules. As said, it is predominant in external secretions not only in the breast milk but also in the saliva, in the tears and the mucus of the bronchial, genitourinary and digestive tract, and hence provide protection at this mucosal membrane level. You know, whether some antigen is going through the bronchial system, through the genitourinary system or through the digestive tract. Any antigen which is passing through any of this tract will be trapped by IgA which is present in the mucous membrane. Now, coming next to immunoglobulin D, it is a membrane bound immunoglobulin found in the B cells. It is less than 1% in the total serum immunoglobulin. Together with IgM, it is the major membrane bound immunoglobulin present on the mature B cells. Although its function is not much known, but it is thought to help in the activation of B lymphocytes within the body. Now, coming to immunoglobulin G or IgG. As I just told you that immunoglobulin G is actually of 4 types: IgG1, G2, G3 and G4. It is one of the most abundant immunoglobulin. It is about 80% the total serum immunoglobulins, and as I just said, it is the antibody or the immunoglobulin present in the secondary response. We all should remember that whenever there is an antigenic challenge, there is a primary response, and if the antigen comes again, there is a secondary response. Now, IgG is involved in producing a secondary response in an individual, and as I just told you in our previous lectures that IgG is produced in large amount in a secondary response. So, it is almost 80% of total serum immunoglobulin. It has 4 human subclasses. One important characteristic of IgG is that it can cross through the placenta. So, actually if we immunize the mother during the gestation period, it will produce more of immunoglobulin, maybe IgG, and this IgG can pass through the placenta into the fetus and give protection to the fetus. So, fetus would be having a better immune system if we immunize the mother during the gestational period. So, IgG can cross placenta also. It is also involved in complement. Now, as I just said that it is of 4 types, IgG1, G2, G3 and G4. Within a serum, the concentration of IgG1 is 66%, G2 is 23%, G3 is 7% and G4 is 4%. Now, generally if you will see the IgG is having a molecular weight of 1,46,000. The sedimentation coefficient is about 7s, and as I have shown, the Ig3 is much bigger than G1, G2 and G4. Now, immunoglobulin E, that is IgGe, is involved in allergic reactions. It is involved in parasitic infections also. It takes care of the parasites. It is very low in concentration unless and until there is an allergen. So, if there is an allergen in the body, the predominant immunoglobulin that will be present would be IgE, its concentration increases. Now, if there is an allergic reaction, it will lead to its manifestation in the form of symptoms of hay fever, asthma, hives, or in an extreme condition it could lead to anaphylactic shock. So, you must remember that the receptor for IgGe is present in certain mast cells and basophils. So, whenever there is an allergen on the second time, this allergen will cross-link with the IgE. The allergen IgE complex will go and cross-link to the IgE receptor present on the mast cells and the basophils. Once this complex has got bound to the mast cell and basophils, it will trigger degranulation within these cells, which will cause to release of certain molecules like histamine, which leads to anaphylactic shock in extreme cases, or in mild cases symptoms like certain hives, asthma. These are diseases or various allergic reactions where there is not too much, but if it is too much of allergic reaction, we call specific term is used, anaphylactic reaction. We will cover this aspect in some other lecture. Now, here what I am showing, this is an allergen which is coming and binding to IgE. The allergen and IgE together binds to the Fc receptor of IgE. The granules present in mast cell undergoing degranulation, and there is a release of histamine and other substances that eventually lead to allergic reactions. Now, there are various variations in immunoglobulin molecules. As I just told you, if the variation in the immunoglobulin is taking place in the constant region, such variation is called isotypic variation. Now, the various heavy chain determinants of various immunoglobulins, mu, delta, gamma, etc. are all isotypes. So, variation in the heavy chain is called isotypic variation. However, if within this constant region at a particular loci there is a variation, that is there is allotypic difference in an allele. Now, that particular allele may or may not present in the two different individuals. So, technically one individual is having one kind of allele while the other individual is having another kind of an allele. For example, in IgG3, there is a variant of IgG3 which is called G3M, which is an allotype which has phenylalanine at position 436 of the heavy chain. So, G3M is an allotype of IgG3. There is another type of variation which is called an idiotypic variation, which is specifically where the variation between two immunoglobulin occurring in the variable region. So, technically one antibody is different from another antibody due to the variable region differences, which is hence they can be called the idiotypes. Now, antibodies, as I just told you, have two major functions: to recognize and bind to the antigen and to induce immune response after binding. The variable region mediates the binding. The affinity for a given antigen is determined by the variable region. It also confers absolute specificity for an antigen. Now, the constant region mediates immune response after binding. Therefore, different classes of constant region generate different isotypes. Now, different isotypes of an antibody have different properties, that is antibody function, which is again dependent on the constant region. Now, coming to the polyclonal antibodies as well as the monoclonal antibodies. Now, whenever there is an antigen coming in our body, it has numerous epitopes. Now, each epitope will have a specific antibody against it. Now, technically an antigen having too many epitopes will be leading to many different types of antibody, where each antibody is capable of binding to a particular epitope. So, technically one antigen, numerous epitopes, different types of antibodies with specificity to each epitope. Therefore, individually one antibody which is produced by one B cell is a monoclonal type, but together they all become polyclonal. So, at any given time in our serum, we have an array of polyclonal antibodies. That means, if we define them, polyclonal antibodies are the antibody preparation from immunized animals. They consist of complex mixture of different antibodies produced by different B cell clones. Now, if one clone of B lymphocyte we are considering, it is producing only one type of antibody and none other. So, technically one B lymphocyte is leading to production of monoclonal antibodies. So, monoclonal antibodies are homogeneous antibody preparation which are technically produced in laboratory. They consist of a single type of antigen binding site produced by a single B cell clone. So, if we consider this, in our body we have an array of monoclonal antibodies produced by different B lymphocytes. Together they form polyclonal antibody sera. If we have to develop this antibody, monoclonal antibody, as such in a human being it is not possible. In any organism we cannot have a monoclonal type of antibody unless there is a tumor of a plasma cell. Otherwise, normally, naturally monoclonal antibodies are not there in the sera. Our sera generally have polyclonal antibodies. But in certain cases there are very many important reasons why we need monoclonal antibodies. So, if we have to produce monoclonal antibodies, we have to go back to our laboratory. Now, here again we are showing what is a polyclonal antibody and what is a monoclonal antibody. Polyclonal antibodies can form lattices with homogeneous monomeric protein antigens, because each antibody can interact with a different epitope of an antigen. While on the other hand, the monoclonal antibodies do not form lattices with homogeneous monomeric proteins, because only they can bind to only one type of epitope on an antigen. So, polyclonal antibodies are a mixture of antibodies with different antigen binding sites that may bind to different epitopes or antigens of the immunizing agent with varying affinities. These are produced by immunizing an animal with the appropriate antigen. A wide array of B cells will hence be stimulated to produce anti-protein antibodies. Now, antibodies may be made to a number of different epitopes of the protein. Even antibodies that bind to the same epitope may have different antigen binding sites and bind the epitope with different affinities. So, all these array of antibodies will together be called polyclonal antibodies. The serum obtained from an immunized animal is referred to as a polyclonal antiserum. It contains antibody to different epitopes and different antigens that were present in the immunizing inoculum. The immunized animal serum is collected. The antibodies are purified from the serum. Since one antigen induces the production of many antibodies, Hence, the result is the polyclonal mixture of antibodies, or the serum is called polyclonal sera. Now, coming to the history of monoclonal antibodies, the monoclonal antibodies were first of all found by the hybridoma technology in 1975 by George Kohler and Caesar Milstein, who devised a method to obtain monoclonal antibodies. They received Nobel Prize for their discovery in 1984. In 1988, Nobel Prize in Medicine was given to Greg Winter for developing the technique to humanize monoclonal antibodies. Now, further in 1986, first monoclonal antibody Murimonab CD3 reached the market, while in 2003, first fully human monoclonal antibody adalimumab was found. Now, these are some of the FDA approved monoclonal antibodies for cancer treatment. For example, Campath, Azemtuzumab, which is used against chronic lymphocytic leukemia, Avastin, which is used against the breast cancer, colon cancer, lung glioblastoma, Erbitux, which is used against the colon cancer, head and neck cancers, Mylotarg, acute myelogenous leukemia, Zevalin, non-Hodgkin's lymphoma, chronic lymphocyte lymphoma, Dutexan, non-Hodgkin lymphoma, Herceptin from breast cancer. This is the sources from the Mayo Clinic. Now, with this I will come to my end. I will like to end my lecture by saying that monoclonal antibodies are extremely important. The reason being, in certain therapeutic, like for example, we just discussed how monoclonal antibodies can be used to combat various kinds of cancers. Now, as such, in nature has a mechanism to give you polyclonal sera only. We will not be able to get a monoclonal sera. It is very difficult to let us say to take out a monoclonal antibody or one type of antibody from a polyclonal sera, because we know polyclonal sera is made up of huge number of different types of antibodies which are specific to different epitopes. Now, to take out one monoclonal antibody and to study it, it is very difficult. Now, we have to devise a technique. Now, question arises, why do we need monoclonal antibody? We have certain, first of all, we want to study immunoglobulins. How will we study? We can only study a particular immunoglobulin by taking it out. Now, as I just told you, it is very difficult to take out monoclonal antibody from a polyclonal sera. So, we have to devise a technique by which monoclonal antibodies can be formed in the laboratory. Now, secondly, to find out or to use this monoclonal antibody for diagnosis. Now, as we know, monoclonal antibody would be very specific to a particular antigen. So, if by some means we can take out a monoclonal antibody which is specific to, let us say, a dengue pathogen or to any pathogen, malarial pathogen or to any pathogen. So, what will happen? We can form a diagnostic kit. Now, we can immobilize the monoclonal antibody on some matrix, and if we put, let us say, a sera, human blood, or let us say urine, and if the antigen is present in any of these body fluids, the antigen will come and bind to the monoclonal antibody present on the immobilized matrix. This is the method of detection in various diagnostic kits. Generally, the reaction is manifested by a color reaction, or sometimes by, for example, a radioimmunoassay is almost always based on this method only. So, for all these methods, whether it is ELISA or whether it is RIA, these are all diagnostic methods where utilization of monoclonal antibodies is taken place. Now, monoclonal antibodies can also have therapeutic use as immunotoxins or as enzymes. Now, immunotoxins are those monoclonal antibodies which are attached to certain toxin molecules. They can go, since they are monoclonal, they will go and bind to a particular antigen within the body, release the toxin there and will kill the antigen there and then itself. So, technically monoclonal antibodies have lot many uses in diagnosis, in imaging, in, let us say, in therapeutics. So, for developing these monoclonal antibodies, we need certain methods. We will come to the method of raising monoclonal antibodies in vivo as well as in vitro in laboratory in our next lecture. Thank you very much.
Okay, on that note, I would like to thank ma'am for this very enriching discussion, and I hope, my dear friends, that you have been benefited by this discussion that we had about antibodies, their structures and their functions, and I hope that you have understand a better understanding of their function and their structures. On that note, I would like to thank you, dear friends, for watching our show. Stay tuned and keep watching. Thank you.